Thanks, Colleen. And before I had a question for Michael, but I saw this one audience question pop up that is related to iPSCs. Corey asked, how often does your clonal iPSC not differentiate to the cell type expected? Kathryn, did you want to?
Sure.
Chew on that a little?
And this is another strength of the bit.bio technology compared to traditional directed differentiation. So in traditional directed differentiation, you're using environmental factors or cytokines or other things to kind of coax the cells into becoming a different cell type. When you're using transcription factors and genomic safe harbors to really kind of do this genomically, it's much more effective. So we have not had any major issues in terms of using different starting materials to generate different cell types. So that's a major difference from directed differentiation for us.